Faculty of Agriculture Technology (UGM), also get doctor in Medical and Health Science Faculty of Medicine (UGM). She also published in several journals. There are more than 38 international journal, more than 10 accredited national journal, 2 book chapters, 4 books of research result and teaching materials, more than 30 IPR, and 3 academic manuscript or draft regulation. She also got research project. There are UGM internal grants and Ministry of Education and Culture grants, and some of them. So the employment history of Dr. Lily Arsanti Lestari from 2033 until present, she is head of the UGM Halal Inspection Institute. So without further ado, Dr. Lily Arsanti Lestari, please welcome. The time is yours.
Yeah, thank you. Good afternoon, everyone, both who join offline in this room and also who join online through Zoom meeting platform. And a very good morning, Dr. Edoardo Capuano.
Good morning.
I'm sorry if there is a mispronunciation of your name.
Not at all. Perfect.
Yeah. My name is Lily. I will be serving as your moderator in this session. And in this session, we will hearing a presentation from Associate Professor Dr. Edoardo Capuano. But maybe before we get started, I will introduce our speaker in this session. I will read a brief of his CV. Yeah. Dr. Edoardo Capuano got his Master of Science from Food Science and Technology, Federico University in Naples, Italy, and his PhD in Food and Health at SUN, Seconda Università di Napoli, Italy. He has worked in several areas of food science and technology, including Maillard reaction and process-induced food toxicant and authentication. Currently, he is associate professor at the Food Quality and Design Group at Wageningen University and Research in the Netherlands. His research interest is in the effect of food structure and composition on bioavailability of nutrient and bioactive compound, as well as on the interaction between dietary compound and gut microbiota. He produced more than 80 articles in peer review journal and also as promoter or co-promoter for more than 10 PhD students. He has so many scientific activities that I cannot read one by one because of the limited time. Without further ado, please welcome Dr. Edoardo Capuano. You will have approximately maybe 90 to 100 minutes, and you can arrange the course. Yeah, maybe you can deliver a 40-minute presentation, and you can add Q&A question and so on. So, Dr. Capuano, the time and screen are yours.
First of all, thank you, Lily, for the nice introduction, and I'm very glad for your invitation to participate in this summer course. Yeah, my presentation is around 50 slides, so I will basically get to my intermediate conclusion before we have a little break. And when I get to the intermediate conclusions, perhaps we can start the Q&A session by asking some questions. I would like to know whether you can see my slides.
Yes, it's perfect.
Okay. So, well, of course, I mean, very good afternoon also to the many students that are attending this course and my lecture from, I understand, all over the world. And I think it's a very interesting topic that this summer course is covering. It's very timely, and it's a very big issue that all of us as humans are facing nowadays. I think it's without questioning that we have a problem as humans. The problems that we have is that we are living too big footstep on the planet due to our activities. And well, the impact of the human activities on the planetary resources is so strong that there are some group of intellectuals, including philosophers, but also geologists and social scientists that are basically they would like to refer to the geological era where we live in as Anthropocene. Anthropos from the ancient Greek means men, humans, to signify that we are living in an era where the human activities are so strong, are so impactful on Earth, so as to also change or affect the geological phenomena, including the climate. I think this is something that is needless to remember. It's out without any discussion. What is actually a little bit less appreciated, especially from the general population, is that the food production chain and basically the way we produce our food, well, it makes a large contribute to the use of the planetary resources. There are many estimates that have been done over the past years, but the most conservative estimates points to the fact that the way we produce our food can contribute to at least 26% of all the emissions of greenhouse gases, which is quite a lot. And it basically points also to a possible solution for the problems we are facing. It's not only about production of greenhouse gases. It's also about use of land, use of fresh water. It's about pollution, the contribution, for instance, of the way we produce our food on eutrophication, but also on the loss of biodiversity, mammal, bird biodiversity. So the contribution of our food production chains is quite large on the use of planetary resources, but not all the food, of course, contribute the same to this loss or waste of resources. And if you look at this graph, I mean, especially if you look at my pointer at this bar, you might basically realize that among the different food that we include in our diet, animal foods, so meat, egg, dairy products, they contribute a lot and most to the carbon footprints. These data are taken from the European Union. And you see that overall, meat, egg, and dairy in total, they contribute more than 80% of the carbon footprints of our diets. Comparatively, plant-based food like cereals or legumes, fruit and vegetables, they contribute much less. And if you look at the bar that is on the right-hand side of the figure, you can also understand that the contribution to the carbon footprint doesn't come much from processing or transport. So the way we process our food, we package our food, or we transport them. But it really depends on the primary production, so a crop cultivation and husbandry. And if we zoom even more further into the different food, we can actually realize that there are some animal products like meats, and especially beef and lamb, that contributed a lot to the global greenhouse gas emissions. These data have been expressed in terms of greenhouse gas emissions equivalents per unit mass of proteins. And you see that beef is the one that by far contributes the most to the greenhouse gas emissions. So we clearly have a problem like the way we produce our food, especially with the way we produce our animal food, has a huge impact on our planet. And this has to be combined with another, let's say, problem we have or situation we have, because Our current population, world population, is well above the 8 billion sapiens, humans on Earth. And the demographic growth is, well, the sapiens are growing on Earth with a very steady pace. And every year there are more humans on Earth corresponding to a country of a large size like France. And according to some projection from the United Nations, we will probably get to 10 billion people by 2050. So 2 billions more than nowadays. And this, of course, poses a problem because, well, for many quarters, there is the understanding that, for instance, within 2050, according to estimates of the Food and Agriculture Organization, because of this demographic growth, because of the fact that developing countries are able to access to level of wealth, increasing level of wealth, that we might need even up to 50% more food and proteins in the future. But it's clear also from the data that I show on the contribution of different food production chains on the use of planetary resources that these current trends in consumptions, in dietary patterns, and in population growth are not sustainable, are not compatible with sustainability. So from many quarters, there is the understanding that we really need to rethink about the way we compose our diets. And particularly, we need to replace part of the animal proteins or totally the animal proteins with high quality proteins that are sourced for more sustainable sources. So we need what is known in the, let's say, in the field as a protein transition. So converting our diet and moving out from the massive use of animal food towards more sustainable sources of protein. One example of such transition is provided by the Eat Lancet report. This is a report that has been produced by the Eat Lancet Commission. It's a commission that gathered a few years ago, bringing together experts in the nutrition, in the food science area, in social science, agriculture. And they pointed out that, yeah, it's possible to switch our diet away from animal proteins. So they proposed a diet, the diet, let's say, of the future, which would be able to respect, let's say, the planetary boundary in terms of resources. And according to this commission, we should reduce a lot the consumption of red meats, sugar, starchy vegetables, increasing correspondingly the consumption of plant-based food. So we need a protein transition. We need to move away from animal proteins, especially meat, because as I showed from some data, meat is the type of food that contributes most to the greenhouse gases emissions, to the use of resources on our planet. So we need actually to find new sources of proteins. So the point is that how to select the proteins to be used in the near future to fuel this protein transition. Well, there are many aspects, of course, that we have to take into account when we select the proteins that we will be using in the future. The first one is, of course, that this protein source should have an ecological footprint that is favorable, or at least is more favorable than animal food. This might sound a little bit of a truism, you know, it's something obvious. But not all the protein sources that have been suggested so far by the scientific community actually they have an ecological footprint that is more favorable than animal products. We will see more about that in the following of my presentation. Of course, I mean, might be a source of protein with high protein yield. That is, for instance, why fruit and vegetables are seldom indicated as possible alternative source of protein, because the yield in proteins is definitely low. It should be something safe, so source of protein that are devoid of microbiological or chemical hazards. Of course, I mean, we're talking about proteins. One aspect of safety is related to allergenicity of proteins. It must be cheap because everybody in the world, anybody in the world should be able to access these protein sources. It must be accepted by the consumers. This is also something very relevant, very much studied by social sciences, because there might be cultural, religious, or even psychological reasons that might, hold people back from using a certain protein source. Next to that, this protein source should also have good technofunctional properties. Technofunctional properties relates to the way the proteins behave in food systems from, let's say, physics point of view. For instance, gelling properties, solubility, foaming properties, emulsifying properties. And this is because, of course, the proteins that we have in our diet do not come only from food, from protein-rich foods, but proteins are also used as an ingredient in many applications. So if we want to replace animal proteins, which have very good technofunctional properties with other sources of proteins, we need to source proteins that have at least decent, adequate technofunctional properties. And of course, last but not least, these proteins must be nutritious. This is where or the protein sources or the protein-rich food that we want to use for the protein transition must provide in the diets adequate nutrients to humans. And this is where I mostly will focus my presentation today. But before moving towards the nutritional aspects of the protein transition, I would like to briefly summarize some aspects of three potential sources of proteins for the future protein transition. So I will tell you a little bit more about legumes, insect, and microorganisms. Of course, the legumes represents what I call the usual suspects, the most obvious candidate to replace animal proteins in the diets of the future. We're talking about the seeds of the leguminous family, technically, botanically, of the Fabaceae family. These are basically very rich in proteins. On average, the legumes contain between 20 and 25% of proteins on dry matter. I'd say most of the 90% of the legumes that are consumed worldwide belongs to a handful of species, beans, lentils, chickpeas, soybeans, peas. But of course, there is many more species that are sometimes grown and known only locally. In Indonesia, for instance, you have jack beans that we have extensively studied also in Wageningen, and that are underutilized and must actually be looked at very carefully for the future protein transition. They are well known and consumed worldwide, so there is no problem in the acceptance by the consumers, and can be consumed as such, but also as protein ingredients. So we can use the legumes as such, but we can also extract the proteins from legumes and use them as a ingredient in many formulations. And one of the, let's say, formulation or the use of legume proteins that is getting some momentum more recently is using legume proteins to produce what we call meat analogs. Meat analogs is a relatively new food concept, is a completely new category of food products that are designed to mimic the texture, the sensorial experience of meat, especially the texture of meat. And well, to produce meat analogs, we need to start from some legumes. The most used one are soybeans and peas. We need to extract the proteins from these legumes, and once we get the protein isolates or concentrate, these might be added with extra protein that are very, let's say, good from a technofunctional point of view, like gluten. And then these proteins are subjected to some sort of extrusion. Different kind of extrusion can be used in this respect, and the twin screw extruder, but also the shear cell extrusion, even 3D printing can be used. Extrusion is necessary to, let's say, change the conformation, typically globular conformation of the storage proteins in legumes to produce the typical fibrous structures of meat. And playing with the extrusion conditions, we can produce fibers with microstructural properties that are different, and so that after adding all kinds of lipids, flavor, and additives, we can produce the vast array of meat analogs that you can find nowadays in the market, from hamburgers to sausages to cold cut or nuggets. So definitely the legume proteins are now very much used to produce this new category of products, meat analogs, that might contribute to the protein transition. The second category that I would like to also discuss today is insects. It is something that is not new to humans, because at the moment there are more than 2 billion humans that practice entomophagy, with more than 2,000 species included in culinary practices all around the world. This is a source of protein with a high protein yield, but depending on the species, depending on the physiological states, whether it is a larva or a pupa or is an adult, the protein content, of course, can vary, but is very often higher than 50% of the dry matter. And from an ecological point of view, insects are an ideal solution, because they can also grow on waste. So they are really perfect for the concept of circular economy. At the moment, there are several species that are used all around the world. In Europe, only 3 species have been, let's say, approved and considered as safe for consumption. So the locust, the cricket, and the larvae of the lesser mealworm. And of course, the insect can be the same as legumes, consumed as such, as a whole insect, but also as ingredients. So and nowadays, especially in the Western countries, we are facing problem in let the consumer accept the use of whole insects. So at the moment, the research is mostly about producing and applying ingredients, protein-rich ingredients from insects in all kinds of food products. And finally, I mean, probably the most novel concept in terms of potential sources of proteins for the future is using microorganisms for that. The idea is to grow cellular biomass in large fermenters, like you see in the image on the right, in what we sometimes refer to as the cell factories. So we grow the cell biomass, and then the proteins are harvested from the cell biomass. Different kind of microorganism can be used for this aim: bacteria, fungi, yeasts, microalgae. And the protein content of the biomass is very variable, and it's very often as high as 70% of the entire biomass. So it's very promising source. And some of the microorganisms are already used by humans since a long time. For instance, microalgae, which is a group of unicellular autotrophic microorganisms, like they can use photosynthesis to produce organic matter. Since a few decades ago, there was also relevant and prominent political figures that were claiming that microalgae might be the solution for our food security problems, like President Obama. Nowadays, we are a little bit more cautious, because more recent estimates have highlighted that the ecological impact of growing microalgae proteins might not be that advantageous as we thought. But anyway, it's a very good source of proteins, with a protein content that is variable depending on the species and the way they are grown, between 10 and 70%, with 3 species at the moment that are considered as safe for consumption in the European Union: the spirulina, the chlorella, and the dunaliella. But also, next to microalgae, also mycoproteins, so the proteins from fungi, are actually on the market since at least 40 years. And we're talking about especially the proteins from this filamentous fungus, which is Fusarium venenatum. which has up to 50% proteins content on a dry matter. Once again, the way these proteins are produced is the fungus is grown in large fermenters, then the biomass is harvested, the proteins are isolated and then extruded to produce. You might have heard about this product, corn. This is a trademark name. You can find it in the market. So it's meat analogs produced based on mycoproteins, or proteins from microscopic fungi. And more, let's say, futuristic or novel solution might come from bacteria. I mean, there is this company that claims that they can produce proteins from hair. And the way they do that is they use bacteria, hydrogen-trophic bacteria, that can literally eat hydrogen, molecular hydrogen. So you grow these bacteria in large fermenters, you provide them carbon dioxide and hydrogen, and they can incorporate it into organic matter producing proteins. And once again, when the biomass is harvested and the protein are isolated, they can be extruded to produce meat analogs. And finally, of course, another possibility is to use genetically modified bacteria to produce the protein of our choice. We can insert genes for instance, animal proteins within the microorganism genome. And then when we grow them in the fermenters, the biomass is growing and the proteins are expressed from the genes that we have inserted into the genome. And for instance, there are many projects all around the world, one also in Wageningen that is funded by the Dutch government to produce dairy proteins without cows. So the idea here is to incorporate the genes for caseins into yeast. So let the yeast grow and develop, and then once the caseins are produced by the yeast, harvest the casein and use it to produce dairy products without cows. And what is actually interesting is that for some of these alternative sources of proteins, let's say, accurate estimates of the ecological impacts have been produced. This is a meta-analysis produced a few years ago where the ecological impact of several, let's say, sources of proteins is compared. Here you see at the moment highlighted the contribution of the ecological impact of legumes. And you see that, as shown also in a few slides above, that the ecological impact of legumes is much, much better and more favorable than beef that you see here on the left-hand side of each panel. This estimate has been done also for insects, and you see that also insect has a very, very favorable ecological impact compared to animal proteins. And also the plant-based meat analogs, that they might have very favorable ecological impact, a little bit less than legumes, because of course to produce plant-based meat analogs, proteins must be extracted from the legumes, which requires, let's say, a little bit of energy and resources. What is also interesting to understand is that such accurate estimates of the ecological impact of other microorganisms has not been done, is not available yet. So we don't know exactly which is the ecological impact of these sources of proteins. But actually, what I would like to stress today is, and it's mostly the topic of my talk, is so we understand that this protein transition is needed. We need to replace meat proteins, animal proteins in general, and meat proteins in particular, with other sources of proteins. So the question is that what can be the consequence, what be the outcome of the result of this replacement on the nutritional quality of the diet. And today I would like to discuss at least 4 different nutrients or aspects of it. So the protein quality, the intake of fiber, iron, and vitamin B12. And we start with the protein quality. As you might, of course, remember, the proteins are made up of amino acids. Some of them can be synthesized by our body. Some of them cannot. We call it essential amino acids, and they have to be provided by the diet. So in a way or another, nutritional quality of proteins refers to the ability that a protein has to provide this essential amino acid to the body. And there are several ways the nutritional quality of proteins can be measured or indicated. The two most important or most used indicators or scores to indicate the nutritional quality of proteins are the PDCAS and the DIAS values. The PDCAS is well, the acronym that stands for Protein Digestibility Corrected Amino Acid Score. And basically, it measures the ability that the protein has to provide, so how much of the essential amino acid the protein is able to provide compared to the ideal amount that is required for growth and maintenance. And so if PDCAS score of a protein is 70%, it means that it can provide only 70% of the essential amino acids that are needed to build our own protein. And the PDCAS score, it depends on the composition in amino acid of the protein, so how much of each individual essential amino acid is present in the primary structure of the protein, and depends on the how much the protein is available to our body, so the true bioavailability. One of the problems of the PDCAS score, as it was defined, is that the values are truncated to at 100%. It doesn't matter whether the protein is able to provide more essential amino acids than ideally required by our body, the values will not be higher than 100%. And another limitation that was highlighted is that the bioavailability of the protein, so how much of the protein is actually digested and absorbed, is measured in the feces. Which is not ideal because it doesn't account for the endogenous nitrogen. It doesn't account for the use of some nitrogen from the microbiota that inhabits the large intestine. For this reason, since 2013, the Food and Agriculture Organization endorsed the use of the digestible indispensable amino acid score to indicate the nutritional quality of protein. The concept is the same. It measures the ability of a protein to provide essential amino acid to the body. So the percentage has to be intended as the extent of essential amino acid that a protein is able to provide compared to the ideal protein, to the ideal requirement of our body. The difference is that the values are not truncated anymore at 100%, and that the bioavailability of the protein is measured at ileum, so at the end of the ileum, which is much more physiologically relevant and realistic. Of course, this is determined by benchmarking the composition of the protein against an ideal proteins, and so we can produce DIAS scores for 3 different age groups. And also typically we consider protein as excellent if the DIAS score is higher than 100%. Good if it's between 75 and 100%, whereas if the DIAS score of the protein is lower than 75%, then no nutritional claim is possible on the package of protein-rich food. And one of the problems we have in terms of protein transition, so replacing animal proteins or meat proteins with other sources of proteins, is that the quality of the meat, but also dairy and egg proteins, is often considered paradigmatic, because the DIAS score of animal protein is often higher than 100%. And basically, recently we have produced a review that we published in the Critical Reviews in Food Science and Nutrition, where we collected the PDCAS and DIAS values for several alternative sources of proteins. And we included in our review also data from animal protein. So in these slides, you see on the left-hand side values of the PDCAS, and on the right-hand side, you see values of the DIAS. These are only data that have been collected from studies in human volunteers or in animal models, in pig models. That is the best model to evaluate the PDCAS or the DIAS scores for proteins. And what you notice is that, first of all, there is a large variability in the PDCAS and the DIAS scores for each protein source. This depends, of course, from the natural variability that you have in biological material, but also depends on the way the food have been obtained, have been processed. But also you notice that on average, the PDCAS and the DIAS scores for meat is higher than any other source of proteins. We have some exceptions. If you look, for instance, at the DIAS scores, you will notice that soybean proteins they have a very high DIAS scores. It's probably the best plant-based source of proteins. The scores is comparable to meat quality. You can notice also in this panel that the plant-based meat analogs can have a very high DS score, especially if they are soy-based, because soy is very high in the DS score. It's also possible that the fungal protein, like mycoproteins that you see here, they have a protein quality that is as high as meat. I haven't reported in this graph the PDCAS values or the DS values for egg and milk proteins, dairy proteins, but it's typically higher than 100%, even 140% for dairy proteins. So the problem that you see is that we have to replace animal proteins that are high quality proteins with other proteins which are less good than animal proteins. One of the reasons for that is that the animal proteins, especially dairy and egg, they are richer in essential amino acids. It is something that we cannot do much about. Another problem is that in general, the alternative source of proteins are less digestible than animal proteins. So you see here, I have collected some data about ileal digestibility of different protein sources, and you see that on average, whereas the meat proteins are very highly and well digested, you see that sometimes the digestibility of other protein sources is lower. You can clearly see the trend, and that the digestibility of the alternative source of proteins is lower compared to meat and other animal proteins. There are two main reasons for this lower bioavailability of the proteins from alternative sources. So in sources like grains, microalgae, fungi, so in all the sources of proteins where cells are surrounded by a cell wall, this cell wall is made up of polysaccharides that are indigestible for humans. They are classified as a dietary fiber. I have some more slides in the following of this presentation. The problem is that since they are not digested, if the cell integrity is retained upon swallowing of the food, then the intracellular nutrients are actually encapsulated within these cell walls. So the digestive enzyme must pass through this barrier to get to their substrate, to the proteins. And this, of course, will slow down the digestibility, the entire process, and reduce the bioavailability of the protein. So every time we ingest a whole grain or whole microalgae with the cell intact, the cell wall are intact, there is a loss of digestibility because of this barrier effect of the cell wall. Of course, I mean, technologically, we can increase the bioavailability of proteins by reducing the particle size or breaking the cells, so applying things like milling or grinding in order to break as many cells as possible and expose the proteins to their enzymes. Another problem is that unlike animal proteins or animal food, plant food and the alternative sources of proteins, they contain a range of what we historically know as anti-nutritional factors, which are factors that can impair the protein digestibility. Things like dietary fiber in legumes, insect and microalgae, or tannins and polyphenols in legumes, proteases inhibitors in legumes and insects, phytates in legumes. Fortunately, we have a set of technological strategies to remove or neutralize this anti-nutritional factor, removing the fiber, isolating proteins, removing the fractions of the food that is richer in fiber and in phenolics, or thermal treatments, for instance, for protease inhibitors that are of protein origin, nature, but also sometimes fermentation and germination, exploiting endogenous or exogenous enzymes to remove, for instance, phytates. But it's important to realize that this loss of digestibility can impair the nutritional quality of protein, and this might have a consequence on the recommended daily intake for protein. So at the moment, the recommended daily intake for proteins for adults is around 0.8 gram per kilogram body weight. But this holds true only if the DS protein of the DS values of the proteins is 100%. So only if the proteins are fully utilizable by your body. But if the protein cannot be fully utilized by the body, the actual recommended daily intake should be actually higher than that. For instance, if the DS value of the protein meal is only 40%, then of course the actual protein intake, recommended daily intake, it should be 2.5 times higher to account for the fact that only a part of the protein can be used. So in this case, it becomes 2 grams per kilogram body weight. And this has consequences on the nutritional status in terms of proteins in the general population. This is a graph that has been produced by Professor Paul Morgan, that is one of the most authoritative scholar in the field of protein nutrition and protein quality. In this graph, you see the intake of proteins in different countries and territories. And you see that the bar that you see is the minimum amount recommended intake for a person of 70 kilos, let's say. And basically what you see from this graph, it apparently that in all the countries, people have enough proteins, which justifies the claims that you sometimes read in the literature that there is no problem in the protein intake. But actually, these data, they assume that the DS score of the proteins in the diet is 100%, but it's actually not. So when you recalculate the actual intake of proteins by including also the DS score in the calculation, what the Professor Paul Morgan did, what you see is that the actual intake of proteins is lower than it should be. So the consequences of having a DS score that is lower than 100% is that the recommended daily intake for proteins is probably or will be probably higher than it's now currently recommended. Which takes me actually to the intermediate take-home message so far. So we have seen that we have a challenge, we have an issue, that the food system and the way we produce our food is unsustainable. The solution for that is that we need to move away from animal proteins, especially meat, beef, towards more sustainable sources of proteins. But one of the possible effects of this transition, of this replacement of animal proteins with other sources of protein, is that the protein quality can be reduced, which actually increases the actually recommended daily intake of proteins. But this can be improved by processing, as we have seen. We have some technological strategies to, for instance, improve at least the digestibility of the alternative source of proteins. But this is, for the moment, my intermediate conclusion. I would rather make a break now, but before the break, perhaps I will ask if there is any question or burning question from the audience that I can answer.
Okay, maybe 2 questions first, yeah. You can mention your name and your question.
Hello, my name is Abu Huraira and I'm from Pakistan. So
Hello.
my question is that as it was a bit described also, but meat protein is generally known as complete source of protein. It has all of required amino acids that our body is required. But like the synthetic protein you were talking about, so how will it compete at that level with I mean the meats, the protein from the meat? And adding into that question, is the protein from plants, fungi, or other microorganisms free from contamination of harmful chemicals, for example, microplastic or metals, or as they can cause metal poisoning also in the body, and as it is highly processed. So, what are the risks associated with it? That's my question. Thank you.
Yeah, okay. There are several questions, and I'm afraid that to address all the questions, we will need another lecture, but it's very good questions. First of all, I would like to make it clear that you talk about synthetic proteins, but why do you think they are synthetic?
Because it's being processed and maybe you can say lab grown. That's why.
Yeah.
I mean, to make it clear between organic and synthetic, I mean that.
Yeah, well, synthetic is when you in the chemical synthesis you produce it by chemical synthesis. This would be the so sometimes, I mean, legumes and insects, there is nothing, let's say, synthetic or lab grown, because they are grown naturally. For the microorganisms, proteins, you're right. I mean, these are basically grown in a lab by fermentation. It's just cellular biomass that is grown in fermenter. I have two slides later about cultured or synthetic meat, but it's at the end of my presentation. But coming back to your question, of course, meat proteins are paradigmatic in terms of quality because they contain, as you said, all and even more than required, all the essential amino acids. That's why the DS values of meat proteins is often higher than 100%. And none of the alternative source of protein is comparable to animal proteins. Soybean is what gets as close as possible to meat proteins and perhaps mycoproteins, so from fungi. But of course, the data we have for fungi is only about Fusarium venenatum, which is the microscopic fungi that is used to produce the quark. So we don't know what is actually the amino acid composition and digestibility of proteins from other species of fungi or other species of bacteria, of yeast. But at the moment, you're right that none of the alternative source of protein is comparable to meat and animal protein in terms of quality. And this might be a problem, as you see from this last slide of mine. When it comes to the contamination, that's, of course, is a very interesting point. I pointed out in one of my slides that the alternative source of protein must be safe, but it's definitely a too broad and a topic to be addressed at the moment. Of course, one of the problems, that's why the authorities all around the world, they are very cautious before granting, the status of safe or grass or whatever to insects, for instance, or microalgae, because because everybody that wants to produce it and commercialize it has to prove that they are safe. So there are concerns, in fact, indeed, you're right. I mean, contamination in the waters where the microalgae are grown, for instance, or contaminations in the feed where insects are grown. There are concerns about that, but there are also solutions to make them microbiologically or chemically safe. And the last thing I think of your question was about processing. I think it's my view as a food scientist, there is a lot of confusion due to the NOVA classification system about highly processed food and whatever. The fact that the protein is processed, the fact that the food is processed is not per se a problem. It's not per se an issue. Processing is not per se producing any harm. Some categories of ultra-processed foods, and we know that since decades, might be harmful from your health because they are energy dense. There are a lot of calories, but processing per se is not a problem. For instance, the meat analogs that I shown, they are highly processed, but there are data clearly shown that they are not harmful for health. They are as nutritious as other, alternatives to meat. And they do not do any harm to, for instance, the gut microbiota or the metabolic responses. So I would be in general cautious in pointing at processed food or high processed food as a source of problems. Does my answer, does my reply answer your questions?
Okay, thank you so much for a very interesting answer.
Okay, next question, maybe. Yeah.
Hello, sir. My name is Gia from the University of the Philippines.
Hi.
And my question is that no matter how many innovations and no matter how we perfect the new technologies as scientists, it won't solve the problem unless it is generally accepted by the public. So my question is that how do we as scientists make our innovations and new technologies more palatable to the public so that it solves the problem that we intend to?
Yeah. Well, this is a very good question. It's very difficult for me to answer this question because this is something that pertains more to social sciences and communication science. I mean, sometimes, as a scientist, we have a problem in communicating our findings to the general public. It's something that is getting worse and worse nowadays with social media and the type of information or communication that is running on social media. I see a lot of, for instance, of misinformation, disinformation, fake news about use of insects. Sometimes this is driven by ideological reasons, political reasons. So I think the point that you raise is extremely relevant because no matter indeed the innovation we do, the same things for the lab-grown meats that I will basically cover in the final 2 slides. No matter how good innovation is, how incredible our achievement in terms of science and technologies are, if the general public doesn't accept it, then it's wasted. And I guess that it takes time, but also takes a different approach from the scientific community, but probably a different way of running the communication nowadays that is mostly through the social media. So personally, I feel that the problem is mostly there. It is something that we as a scientist can do, but not alone. I mean, we need also political actions there.
Yes, I do understand what you said. Like the public are growing increasingly more suspicious and hateful of the science community, and the science community are growing more distant. And it seems like the science community is at an echo chamber, like we're talking to each other, not really reaching the grassroots. And I think that one thing we can do is that we shouldn't rely on like middlemen, middle media to communicate what we want to the public. And instead, as scientists, we should um, put ourselves to the levels of the general public so that they would maybe hate us less and maybe we can see things, we can see ourselves from how they see us. I think that's something we
Yeah,
can work on.
it's a respectable opinion. I honestly I don't have a solution for that because you really need a probably social scientist to answer this question. But the way you say, I mean, the middleman can, I mean, if the public is trusting scientists, then getting the middleman away might be a solution. But does the general public trust the scientists? I get the impression that the trust in science and scientists is less and less nowadays. That's why perhaps the media or intermediate men that are trusted by the general public might be needed to transfer the message. I don't know. I mean, that's really something related to social science, but it's a very good point. It's something that might be a really big issue in the future. I agree with
Okay,
you.
thank you, Doctor. I really liked the presentation today.
Maybe one question from the online participant, if any. No, maybe I have one question for you. About the safety of alternative protein source, especially from insect. Is there any toxic substance or allergic compound found in the insect?
Yeah, well, it's also a good question. That this is something that I typically do not cover because it's not, let's say, really the core of my expertise. I don't think there are endogenous toxins in insects. There might be environmental toxins depending on how the insects are grown, because it is claimed that they are good for circular economy because they can be grown on waste. But of course, growing it on waste means also exposing it to potential environmental contaminants. And there is an issue about probably allergenicity because of cross-reactivity with crustacean proteins. And there are several actually projects in Wageningen from colleagues that are investigating the allergenicity of insect proteins. So there might be an issue there, but at the moment I don't have data to, let's say, support any firm conclusion about that.
Thank you for your answer. Maybe you can proceed to the next presentation.
Perhaps, I mean, we can have a 5 minutes break as we
Oh, okay,
agreed.
okay.
And I will
Yeah,
be back
okay.
in 5 minutes and I will continue with the rest of my presentation.
Okay. Yeah, we will have 5-minute breaks and
Yeah.
then we will
Thank you.
continue with Dr. Capuano presentation.
Thank you. Hi. Lily, I'm ready to start. Give me you give me the signal where to start.
Yeah, we will start the lecture again.
Yeah. Okay. So thank you for the break. I really needed to rehydrate myself. So basically, before the break, we left with this intermediate message, like one possible effect of the transition, the protein transition, is loss of quality of protein, and so that the actual recommended daily intake of proteins can be higher than it is at the moment. Of course, I mean, when we consider the nutritional quality of the diet, it's not only about proteins, but of course, the diet, a protein transition implies that protein-rich animal food are replaced by protein-rich sources of proteins from other sources, right? More ecologically favorable sources. So we have to look also at other nutrients that are present in the diet next to proteins. And this might be an issue as has been highlighted by this study that has been published in the Lancet Plant Health last year about some calculation that some scholars have done on the Eat Lancet diet. Before the break, I mentioned that the Eat Lancet Commission, they came up with a diet, a possible solution to the protein transition, how we should change our dietary pattern in the future, remaining within the boundaries of the planetary boundaries in terms of resources. Less meat, more plant-based food. But basically, when the scholars have made a calculation about the amount of nutrients, of micronutrients provided by this new diet, they realized that there are some of these nutrients like vitamin B12, calcium, iron, and zinc, which intake would be lower when we implement this protein transition, if we would like to use the diet proposed by the Eat Lancet Commission. So switching from animal to other sources of proteins might compromise the nutritional status in terms of other nutrients, other micronutrients, vitamins, or minerals. And today I would like to actually address especially three things. So the intake of fiber, the intake of iron, and the intake of vitamin B12. And I will, of course, I mean, again, I will look at the different alternative source of proteins. In this second part, I will only focus on some of them, so soy pea, microalgae, insect, fungi, and plant-based meat analogs. And I will start with the dietary fiber, which you might remember the definition of it. It's, well, according to most of the international authorities, national and international authorities, fiber is a group of carbohydrate polymers with more than 10 monomeric units, which cannot be digested by the human enzymes in the small intestine. So they flow into the large intestine. We can refer to dietary fiber when we have these carbohydrate polymers that are naturally occurring in their natural source, which is mostly plant, whether they are extracted from natural sources, but they have a proven health effect, or even obtained by synthesis with proven health effect. And well, the definition of fiber can or cannot include carbohydrates with more than 3 and lower than 10 monomeric units, but it is left to the individual authorities, national or international authorities, whether to include these oligomers in the definition of fiber. And the current recommendation for fiber intake in adults is quite variable, but typically is from 25 to 35 grams per day. And the problem is that, especially in the Western countries, we can barely reach this recommended daily intake. So the actual intake of fiber is lower, sometimes it's much lower than the recommended daily intake, let's say 30, 35 grams per day in US or 25, 32 in European countries. And from this respect, the protein transition might be an opportunity because by definition of fiber, the animal proteins, animal food, so meat, dairy, and eggs do not contain any fiber. Whereas alternative sources of proteins do contain some fiber. In this graph, I have made some calculation and I have reported that the gram of fiber that you have for each gram of proteins in all the alternative sources that I'm covering in this second half of my presentation. And you see that it's the amount of fiber per gram of protein is quite variable, but there are some sources like pea or microalgae that are quite rich in fiber. So I did also a sort of exercises, and I actually was wondering what would be the contribution in terms of fiber to our diet when we replaced part of whole of the meat proteins with each alternative source of protein. So what you see in the right-hand side of the slide in this graph is what you see here is the contribution in terms of the daily contribution in terms of fiber if we replace 50% of the meat proteins with each of the alternative protein source that you see here, or if we replace completely the meats with these protein sources. And what you see here is that if the protein transition is implemented, and especially if all the meat proteins are replaced, then the contribution in terms of fiber is substantial. Well, if theoretically, if all the meat proteins are replaced by microalgae or pea protein, then you might easily reach the recommended daily intake for men in US and Europe, which is represented by this horizontal bar. So it will be already sufficient to provide all the fiber that is recommended for our daily diet. But of course, I mean, we also have to acknowledge that, well, we refer to fiber as a sort of one class of compound, but in fact, fiber is composed of molecules with a large chemical heterogeneity. So when we talk about fiber, we refer to things like oligosaccharides, like fruit oligosaccharides, galacto-oligosaccharides, inulin, but also large high molecular weight polymers like resistant starch or non-starch polysaccharides. Lignin, which is not a carbohydrate, but it's made up of phenolics, is also included in the definition of fiber. But the point that I want to make is that sometimes we consider fiber as a, say, uniform category in terms of physiological effect, but this is not the case, because indeed fibers of different types, they differ in terms of sugar composition, types of linkages, number of side chains, distribution of side chains. So all these molecular chemical differences, they translate in a differential interaction with water within our gastrointestinal tract. And it changes the physical properties of the fiber, like solubility, water holding capacity, the capacity of forming gels or viscosity, capacity to bind or absorb other nutrients, the fermentability of the fiber in the large intestine. And it's this that determines the physiological effect. So the point that I want to make is that we cannot claim that all the fiber has the same physiological effect, but the physiological effect really depends on the physical properties on the fiber, which in turn depend on its fine molecular properties. And when it comes to the physiological effect of fibers, I remind you that there are at least 4 different physiological effects of fibers. The first effect is what I have described before the break in terms of digestibility of proteins, that when the fiber is still structured within the intact cell wall, it might form a physical barrier for the enzymes to reach their substrate, which modulates the nutrients bioavailability, reducing, for instance, the caloric intake, but also limiting the bioavailability of proteins. There are some fibers that, especially high molecular weight, soluble and poorly branched fiber that are able to form a very viscous solution within our gastrointestinal tract, which can help regulating the baseline cholesterol level or the postprandial glycemia, which are all beneficial effects. Some fibers can also bind endogenous and dietary components like bile acids or minerals or phenolics, which might modulate their bioavailability and in turn also the metabolic responses. And finally, all the fibers that reach the large intestine, they can be fermented by the gut microbiota, producing short-chain fatty acids that are beneficial for gut and systemic health. And so the physiological effect of fiber really depends on the type of fiber we have in our diet. So we cannot claim that the fiber that is contributed by alternative sources of proteins will have the same physiological effect. In fact, the fiber composition of different, let's say, alternative sources of protein is quite diverse and different. So legumes like soybean and pea that you see here are, for instance, rich in hemicellulose, pectin-resistant starch. They are highly fermentable fibers, so they are probably beneficial for the gut health and might modulate the postprandial glucose metabolic responses, like the postprandial glucose peak in the blood. Comparatively, the fiber that are present in the microalgae also contain hemicellulose and pectin, but also alginate and agar. These are less fermentable fiber and less able to form viscous solution. On the other hand, plant-based meat analogs may have a composition of fiber that is similar to the plant source of proteins, which is soy or pea. But during the isolation of the proteins or the fractionation process, some of the fiber types can be lost. So the composition is not exactly the same as the legumes. When it comes to insects, for instance, the only source of the only type of fiber you have in insect is chitin, which is a fiber that is that the insect use to make their exoskeleton. And the chitin is a poorly fermentable fiber, and it's not able to produce viscous solution. So the point I'm making is that it's true that replacing meat proteins with other sources of proteins can contribute even substantially to the amount of fiber in our diet, but it's difficult to judge what can be the physiological effects of this replacement because the type of fiber that are present in different sources is rather different. So I don't expect, for instance, that the fiber provided by the insects can improve gut health better than legumes because the chitin is very little fermentable. So the effect of the protein transition might be beneficial in terms of amount of fiber that is provided in our diet. The situation is definitely different for iron. You might, of course, remember that iron intake is crucial for humans because iron is a metal cofactor in many enzymes involved in all kinds of metabolic activities, oxygen transport, energy metabolism, immune function, DNA synthesis, and also others, cognitive and neurological development. The deficiency of iron can cause quite serious physiological conditions, anemia, neurological dysfunctions, impairment of the immune system. Sometimes even too high level of ferritin in the blood can be harmful. There are some indications that, for instance, the level of ferritin in the blood is positively and significantly associated with the metabolic syndrome. So even too much iron in the diet can be harmful. I remind you that the recommended daily allowance for men and post-menopausal women is around 8 milligrams per day, which is even higher for premenopausal women for obvious reasons. And when we look at the level of iron present in the alternative source of proteins, it's quite surprising to see that many alternative sources of proteins, like plant-based meat analogs or soy or insects or microalgae, might even contain more iron than meat. And so this is potentially something very positive. But there is a caveat here that is necessary to address, is that not all the forms of iron that we find in food or dietary iron are equal in terms of efficacy. Typically, we have two different forms of iron, so the heme iron and not-heme iron. The heme iron is iron that is coordinated to this large molecule, the heme. This is typically present in meats, in hemoglobin and myoglobin, because the heme is the prosthetic group of myoglobin and hemoglobin that binds oxygen. So the iron that is coordinated to the heme iron is present in a very soluble form and cannot be chelated by other dietary components, which makes heme iron present in meats highly bioavailable. Whereas the iron that is present in, say, not heme form, this is present in the more insoluble form and can be chelated by other dietary components like phytates or phenolics, which will reduce the absorption of iron. So end of the story is that the heme iron that is present in meat is much more bioavailable than not heme iron that is present in plants, for instance. And we know that vegetarians, for instance, are more likely to develop iron deficiencies compared to non-vegetarians because of the much lower bioavailability of iron in plant-based food. So the point I want to make is that even though the intake of iron can be higher when we replace meat with other sources of protein, this doesn't mean that the iron status would be as good as when the meat or animal proteins are included in the diet. And this is because the iron that is present in all the alternative forms of proteins except insect, partially in insect, is in a non-heme form. And also these alternative sources of proteins, they contain also other dietary components like phytates and phenolics that can compromise the absorption of iron by chelating it. So an exception can be insects, because we know that at least part of the iron in insects, which is animal, of course, is in the heme form. And finally, I mean, there is the vitamin B12. This is also an essential vitamin for our health. It's essential for DNA synthesis, the maturation of the red blood cells, the functioning of the nervous system. And also for the vitamin B12, like for the iron, we might have a problem in the protein transition because vitamin B12 is only present in animal products and in meat. So we know already, for instance, that vegans and vegetarians that exclude meats, they have lower intake of vitamin B12 compared to omnivorous subjects. And when we look at the level of vitamin B12 in different alternative sources of proteins, we see that for many of them, like for instance, legumes or mycoproteins or plant-based meat analogs, it's completely absent. It's present, but in an inactive form in microalgae. It's present mostly in an inactive form in insects. So we can foresee that one of the problems in the protein transition, replacing part of the meat proteins with other sources of proteins, is the vitamin B12, because meat is the only high rich source of vitamin B12. A possible contribution can be from insects, but more research is needed to understand the contribution of the inactive forms on the vitamin B12 content of insects. And the final two slides that I have, I wanted also to briefly touch, without going much into details, about cultured meat, which is also known as cultivated meat or synthetic meat, in vitro meat, whatever you want to call it. It's a new concept. It's very futuristic. it's a completely different concept compared to the meat analogs. In meat analogs, we take proteins from different sources, plant, insects, microorganisms, and we structure them in the form of fibers by using extrusion. The cultivated meat, the cultured meat, is actually based on different techniques that have been pioneered in regenerative medicine, in tissue engineering. The idea is to take a biopsy from a living animal and to grow the actual animal cells in a fermenter, allowing them to structure themselves in the tissue as they are in nature, by using specific growth factors or scaffolding material to orient the fibers in the perfect way. Some consider that's the solution for the protein transition, because you can ideally produce high quality proteins in a sustainable way, and cruelty free, so without any animal suffering. Truth is that we don't have any accurate accounts or estimate of what is the ecological impact of growing cultivated meat. So we are still waiting for that. And also from a nutritional point of view, we know very little about that. So in theory, you have proteins that should have the same nutritional quality as meat proteins. But in fact, we don't know exactly which proteins are expressed and developed and grown when you cultivate meat. We know, for instance, that only neonatal or embryonic form of actin and myosin are developed in vitro meat. So we don't know which is the actual essential amino acid profile of these proteins, nor we know the digestibility of these proteins. What we know is that this type of meats can provide some fiber, a little bit, from the scaffolding material, which is this 3D structure that is used to grow the cells on, which is typically made up of cellulose or chitosan, which is a derivative of chitin. We also know that this is a meat that will not contain any iron or vitamins unless we provide it in the growth medium. So there is no way this meat can synthesize vitamins B12. And we don't even know whether the iron provided in the growth medium would be uptaken, so would be bioavailable. So it's possible that actually this type of meats would not provide iron and vitamin B12 as it was supposed. And this basically is my last slide, so I would like to conclude it with let's say the full take-home message. Again, I mean, the challenge we are facing nowadays it can be solved by implementing a protein transition. We have seen before the break that the replacement, partial or complete, of animal proteins, meat proteins, with other sources of proteins that are more ecologically sourced can reduce the quality of the protein, but can also increase the intake of fiber, which is definitely beneficial given the fact that for most of the countries, the intake of fiber is well below the recommended daily intake. But there might be also a problem in terms of iron intake or iron status, because most of the alternative source of proteins, they might have high level of iron, but this iron is not completely bioavailable. And definitely there would be a problem in terms of vitamin B12, because meat is the only rich source of vitamin B12. So we will need to supplement vitamin B12 in the alternative source of proteins that we will use in our future diets. That is basically my final slides. Thank you.
Okay, thank you. I will open the Q&A session. Is there any question? Okay.
Yes, my name is Abu Huraira and I'm from Pakistan. And my question is that regarding the second last slide that you just showed, and where you were talking about the effects of the transition of protein. And don't you think that there can be one more effect that would be increase intake of the preservatives or possibly some chemicals that might be harmful. Don't you think this can be possibly an effect also? That's my question.
Yeah. Yeah, once again, indeed, I mean, one of the aspects we have to consider next to ecological considerations and costs, cultural, consumer, cultural aspects is indeed the safety. I agree with you. I mean, it's not that the animal proteins that we consume nowadays are completely free of hazards, microbiological or chemical or physical. So it's just a matter of designing our production chains of the alternative source of protein in a way that they are safe. You mentioned preservatives or additives, but I would like to stress that the same way I did for the ultra processing idea. The way the ultra-processed food are defined according to the NOVA classification is that the food that are ultra-processed and contains additives, that contains preservatives. But we know that because it's decades that we investigate that preservatives and additives per se are not a problem. Otherwise, the authorities all around the world will not approve their use, and they would not consider their use safe for human consumption. So personally, I don't see any problem at all in the fact that additives or preservatives are present perhaps in higher concentration when we replace animal proteins with another source of proteins. Different, of course, the problem might be different if we refer to some chemical others or some toxin that are naturally present in the alternative source of proteins. This is something that definitely is a problem, might be a problem.
Okay, thank you for the answer. I was very concerned about this because a few reasons. One of the main reason is that because when we were transitioning from like the old packaging materials like tin cans and glass bottles to plastic, then at that time it was like promoted and it quickly like gained went to the local consumers. But then now we conclude that plastics have harmful BPAs and other microplastics that go and penetrate our bloods and our body system, and it can be harmful for us, especially like increase estrogen and this stuff. So that's the reason I'm very concerned about this effect of the transition.
Yeah.
So
No, I get your point, and it's definitely you're right. I mean, several decades ago, we couldn't foresee that the microplastics would be a huge problem. So I think that if you think about that, it is also something related to the nature of the truths that we scientists can uncover. So there is no absolute truth in science. We can never claim in science that something is absolutely true. That will never change. It's not in the nature of the type of, from an epistemological point of view, we can never claim that we have got the final truth. There is always space to change our minds. There's always space to change our theories. And this applies also to the considerations we make about what is safe or not. So it's true that it might be that nowadays we don't see a problem that we will face in 20 years. But I remind you that 40, 50, 60 years ago, also the consumption of animal proteins was advertised as extremely healthy. And nowadays we know that it's not.
Mhm.
So, I mean, 100 years ago, animal proteins were the ideal best, right? But nowadays we know, and our view has changed in terms of animal proteins. They are necessary in small amounts, but we know what is the harmful consequences of consuming too much animal proteins, which could not be seen after the Second World War. So it's true that you are absolutely true. We might have problems that we cannot see at the moment. This is very very little we can do about that. But the same might apply also to the animal proteins that we want to remove from our diets. We might discover in 20 years' time that actually meat proteins are even more harmful than we think nowadays because of something that we cannot see now.
Hmm. Interesting. So if we conclude that from your statement, if we conclude, would it be right or wrong that just like we mentioned that we're not sure about the safety. There might be many side effects that we are not sure about right now. So just like there are cigarettes which are being sold, and but there's written on the cigarettes the effect of the cigarette. So why don't we present these products like it's still in the testing phases? It might have harmful effects on the health, so that the consumers might know rather than saying it might be safe. What do you think if we conclude?
No, but,
Would it be right or wrong?
no, but I mean, what I mean is we cannot foresee a problem, but we have strict rules to approve products, to put products on the market that are safe. It took 10 years before the insects could be marketed in Europe to get the approval from the European food safety authorities, because the companies that wanted to market the insect ingredients had to provide all kind of proofs, all kind of data proving that the products are safe. So the procedure to approve as a safe something in, at least in Europe, it's extremely long and cautious. It's really designed to protect the consumers. So I think most of the cases we do all we can to test and to prove that something is safe. Clearly, as you pointed out, there might be things that we cannot see at the moment. But the procedures that we have when we have in place, they are very strict. So, I mean, something that is still under testing will never be put on the market in Europe. Of course, different countries and territories, they have different regulations. In the US, probably the market is a little bit more liberal, so they are a little bit more free, but still, even the FDA, they requires proofs that something that is you're exposing the public to is safe for consumption.
Hmm.
So there is no way that something that is not tested or where is still a pending question mark is put on the market, at least not in Europe. Not at all.
Hmm. Okay, I got the point. And thank you so much. I really appreciate the answers. The presentation was great and very brief answers. Thank you so much.
Thank you. Thank you very much.
Is there any question? One more, maybe? No? From the Zoom meeting? Yeah. From the online participant, yeah. We have another question, but I cannot read because it's too small.
It's something in the chat, in the
Chat, yeah, chat room.
let's see.
Can you read the?
Yeah, I cannot
I cannot
access.
read because it's too small for me.
Um,
Oh, question from
Oh, so I see if it's true that when we cook animals alive like crabs, we intake their pain and could lead to lifestyle illness like cancer.
oh, yeah.
Is this the question that you were referring to? I see two questions. So the first one, if whether the animal suffering can actually affect our health. Well, I would say no. I would say there is no link. There is no link that is supported by any scientific data that animal suffering and the biochemical effect of animal suffering can somehow affect our health. Animal suffering is a problem per se. My personal view is it is unfair. So we have to do something to reduce as much as possible animal suffering, whatever it takes. But just because of the animal welfare per se, but not because it's harmful for our health. And the second question that I see in the chat is about how food system would affect climate change in general, and which would be the best way to select proteins, which would be the best for human consumption. This is a little bit what I've discussed especially in the first part of my presentation, especially the crop cultivation and husbandry that impact our planet. It's not as much the way we process our food or we transport them or we package them, but the way we produce the primary production. So this is the way at the place we have to act on the primary production. And when you see the data that I showed in the slide about the individual contribution of different foods, the greenhouse gas emission, is clear that the problem is mostly in the animal proteins and the problem is mostly in the beef production, if you look at the data. So if we want to have the highest impact in terms of reducing the greenhouse gas emissions, we have to cut the consumption of animal products, beef, meat products, and beef, especially.
Okay, thank you for your answer. Because of the time limitation, I'm afraid I will end this session. Thank you very much, Dr. Edoardo Capuano, for addressing this interesting topic.
Well, I thank you very much. I mean, I'm very glad to have been here and presenting, and thank you very much for the invitation for this nice initiative. I would like to also thank the people there and for the questions. They're all really, really great questions, very good points. And if anyone that has attended this presentation online or in presence there, they have any additional question they would like to ask me, they feel free to reach me through my email address. I would be happy to answer your questions.
Yeah, okay. On behalf of the summer course, I'm sorry. On behalf of the summer course committee, we would like
Not hear you anymore, Lily.
I'm sorry for the trouble.
Okay.
Yeah. On behalf of the summer course committee, we would like to give you a certificate of appreciation. Please accept this certificate as the speaker of the summer course. We hope we can see you again in another course or conference. Thank you, Dr.
Hopefully
Capuano.
in person next time.
Okay. And then I will give back the time to the Master of Ceremony. Thank you.
Thank you very much. Bye-bye.
Thank you again to Dr. Edoardo Capuano for insight, also sharing the topic about protein transition and the nutritional quality of the diet. Also, our moderator who lead the discussion before, Dr. Lily Arsanti Lestari. So the next, we will give a certificate appreciation to Dr. Lily Arsanti Lestari. The certificate will be given by Dr. Viameta Ayu Purwandari as the committee of this summer course program. So, so don't forget to fill the attendance form by scan the QR code on the screen, please. The operator help me to share it. To Dr. Edoardo Capuano, if you have another activity, you allowed to leave this Zoom session. Thank you. So we would like to remind all the participants for the group task. Also, we also want to remind to all the participants that there are post tests that can be accessed in ELOC platform. So don't forget to complete the post test. And of course, there are tomorrow's schedule, which is a field trip. Please pay attention to the rules and make sure to bring your necessary.
Okay, everyone. Thank you for your enthusiasm for today's course. Tomorrow we will have another field trip at the Center of Agro-Technology Innovation in UGM. It's not so far if we compare to yesterday, maybe 30 minutes by bus. So in the field trip, you will learn about the waste management system that being operated by the UGM. So the for the rules for tomorrow, it's not so different with yesterday. Please uh wear your hat or cap, and then the uniform or the dress code is up to you. It's free now. You can use your t-shirt before, or you have another clothes. It's up to you. But uh yeah, okay, that's and then the other one is please wear your sunscreen. Also bring your bottle of water. We will provide you the uh, water for the refill. And we will start going to the location at 7:30. So please make sure that you come on time at 7:30 in this faculty.